Hi, I'm Clara Fuchsman at the Horn Point Laboratory at the University of Maryland Center for Environmental Science. And I'm going to talk to you today about contributions of vertically migrating zooplankton to sinking and suspended particle matter fuel N2 production in the ETMP oxygen deficient zone. And I just wanted to take a moment to acknowledge my co-authors. And I'm just going to say that I'm invited to give this talk because we just had our paper published in GBC. Okay, so getting started. There are 3 natural marine oxygen deficient zones in the ocean where the definition of an oxygen deficient zone is less than 10 nanomolar oxygen. There's the Eastern Tropical North Pacific, which is where we're going to be mostly talking about today. There's also the east off of Mexico. There's also the Eastern Tropical South Pacific off of Chile and the Arabian Sea by India. And oxygen deficient zones are a mid-water feature. So here is the oxygen profile. You can see the dashed lines are the top and bottom of the oxygen deficient zone, and that it increases again at depth. So the ETMP, oxygen deficient zone, has been expanding both vertically and horizontally. So why does that matter? It matters because oxygen deficient zones have a global impact on the nitrogen cycle. Here's a really simple nitrogen cycle where you have nutrients in this box. These nutrients are essential for phytoplankton and bacterial growth. The input here is nitrogen fixation and the output is N2 production. But 30 to 50% of marine N2 production occurs in oxygen deficient zones, which are only about 1% of the ocean volume. So organic matter is necessary for N2 production, and actually both N2 production pathways need organic carbon in oxygen deficient zones. Heterotrophic denitrification is obvious because it uses organic matter. But Anammox, which is an autotrophic process, needs ammonia. And in most of if you get away from the coast, there's basically ammonia is unmeasurable in the water column. So it needs organic matter degradation to produce ammonia. So here is satellite imaging of the Eastern Tropical North Pacific, and most of the ETMP is offshore with low chlorophyll. So this black line is the oxygen deficient zone, and you can see that most of it is in surrounding blue water. And productivity offshore is really low, and sediment track fluxes, they're all at oligotrophic levels. So that we're looking offshore here at the offshore oxygen deficient zone, and the maximum in the maximum biological N2 gas in ETMP at our favorite station is in the core of the oxygen deficient zone. So here we have biological nitrogen gas concentrations that have had the background abiotic nitrogen gas removed, and we have 2 peaks, one in the top, which is and then we have one in the center of the oxygen deficient zone, about 300 meters, which is exactly the same range where ADC backscattering indicates migrating animals. So zooplankton are tiny animals in the ocean. They undergo daily vertical migration to avoid predators. They transport organic matter from the surface to the deep. So this is just a picture of that. We've got our copepod. It's eating algae at the surface. It could be eaten by fish. So it swims down into the darkness to avoid the fish during the day, and it poops and pees at depth, and this is food for bacteria. And we can see the zooplankton migration looking at backscattering data. In this particular example here, we have the day in red and the night in black. And what you can see during the day is there's a maximum, and there's the most peak maximum is at 270 meters, and this 270 meters is going to keep coming up over and over again. But these backscattering techniques, they see large zooplankton. Obviously, no zooplankton are really large, but such as lanternfish, krill, pteropods. It can't see small copepods or other small zooplankton. Okay, so we had these NSF Dimensions cruises to the ETMP. I was a postdoc on this grant and then went to later cruises as a professor. The point of these cruises was to get detailed organic matter flux data from sediment traps and in situ N2 production rates. The zooplankton angle was a surprise, though not a completely surprise because we had read Daniele Bianchi's modeling paper about how zooplankton could affect oxygen deficient zone. So we were looking, but it was not a zooplankton cruise. It was an organic matter cruise. Okay, so the question I'm going to talk about today are zooplankton adding organic matter and ammonia at their vertical migration depths in the oxygen deficient zone? To answer this question, we will examine quantitative qPCR of metazoan, which is like the official name for zooplankton and forage fish, DNA. We're going to look at ammonia concentrations, suspended particulate organic matter composition, sinking particulate organic matter composition, sinking carcasses, and N2 production rates. So if we look at the quantitative qPCR of metazoan eDNA, we'll just take a moment to say what eDNA is. eDNA is the DNA in the water that is no longer connected to the organism. So this is just filtered water that we then looked at DNA for metazoans, so zooplankton and fish and others. And okay, so the maximum of the metazoan DNA, so the dashed lines are the oxygen deficient zone, and the gray lines are where the zooplankton migrate to, their migration range. And here I've marked this 270 meters, and there are these maxima in metazoan DNA in this 270-meter region. And the error bars here are biological replicates, so different Niskin bottles. But you can see it happened repeatedly on different days. And there's other slightly elevated values also in this region of vertical migration. So this kind of shows, like we saw from backscattering, we think that they were animals, and then we're like, okay, we've got our DNA. They're definitely animals in this region with a peak at 270. So now we're going to look at ammonia. And we see also a maximum here in the vertical migration range, particularly the biggest value is huge. It's absolutely huge value. So these are OPA ammonia data here, and dashed lines oxygen deficient zone, and this blue square this time is the depth of vertical migration, and we're using OPA ammonia, so the detection limit is around 10 nanomolar. So most of these values are, they're a couple, often they're less than 100 nanomolar. And then we have this one that's at 2 micromolar, 2.5 micromolar. So it's just these huge value, and also these, but it doesn't happen all the time. So we would say that zooplankton are likely adding ammonia to the oxygen deficient zone. This is logically possible because to withstand anoxia, zooplankton use anaerobic pathways, and they also use repressed respiration, and they excrete less under anoxia, but not zero. They're still alive. So they are in the oxygen deficient zone, probably excreting ammonia. Now we're going to look at organic matter, and there's some helpful information for examining organic matter, which is that carbon to nitrogen ratios are lower for zooplankton than algae or degraded material. That means zooplankton, their bodies, and also their fecal pellets. That means they have more nitrogen. And the other thing is that zooplankton are enriched in C13, like just a bit, compared to photosynthesizers, and degraded organic matter, it doesn't become enriched. It either does nothing or it is slightly more depleted. So zooplankton are slightly enriched and they have more nitrogen. Okay, so that's what we're looking for in our system, and not surprising, that's what we see here. So here we're going to see a low C to N ratio of suspended. So this is on a GFF filter of particulate organic matter at the backscattering maximum. So in blue right here, we have particulate organic matter. And here we can see our 270-meter depth. And we have our C to N ratio that has its smallest value. So low C to N ratio at 270 meters. At the same time, we have this tertiary nitrite maximum. Well, it's really more of a shoulder on the nitrite peak, but we see it over and over and over again. So we think that this shoulder is real, and it's also in the same range as these low C to N ratios. And I wanted to note that this is kind of at a small depth range. It's not over the whole range where we saw backscattering. And my thought about this is just hypothesis, is back in the 80s when the Russians were looking at zooplankton in the Black Sea, they looked through submarines and they could just see out the windows with a microscopic window somehow that the zooplankton were migrating to thin layers of different species. So I'm wondering if something like that is happening here, but I don't have any proof of that. Okay, so we've got low C to N ratios, and then when we move on, this is the same C to N ratios peak at 270, and we look at C13, which is here in purple, we see enriched C13, or so it's less negative. So the enriched C13 is also possibly indicating of zooplankton input. So this is suspended organic matter. And then if we look in the sinking organic matter, we see exactly the same signals. If anything, they're stronger. So if we look here at A, we have just sinking particulate organic matter fluxes. So we have a lot of sediment trap data. And we also have shells, particularly these are pteropod shells. So you can see that there's a maximum of pteropod shells at 270 meters. And we have increased particulate organic nitrogen. So our C to N ratio is also low, which could be indicating zooplankton. And our C13 is enriched at 270 meters in the sinking organic matter. So we see it both in the suspended and the sinking. And at the same time that we had organic matter data, we also could look at carcasses from the sediment traps. So how do we do that? To determine organic carbon in sinking carcasses. So these sediment traps were different than normal in that they had no poison because we're also using them to do in situ N2 production rates, which I'll talk about at the end. There's time. So the carcasses are real carcasses. They're not poisoned. But all the time, carcasses are always removed from sediment flux data. You can't publish unless you remove them. So we removed them, but we took photos first. We took photos with the macro lens, and we got rid of them. But then we could go back in time and determine their length and width with image J from the photos and convert to organic carbon. And so we could still manage to calculate organic carbon of these zooplankton. And so here, this is the same sediment trap data that we've been looking at. So the red circles are particles, normal aggregates, and then in these squares, it's these carcass data. And you can see there's just a huge amount of carcasses at this 270-meter depth. And they are, well, pteropods and euphausids, a lot of them. So we found 3 euphausids, 19 pteropod shells. Some of them had bodies, some of them didn't. 2 amphipods and 1 cnidaria, so jellyfish. Well, that gives you an idea of kind of who's there a bit. Certainly, there are pteropods and euphausids in this depth range. Okay, so in summary, the vertical migration maximum of 270 meters is a hot spot. At 270 meters, migrating zooplankton add organic nitrogen to the system. And this organic matter is used by microbes to reduce nitrate to nitrite and produce N2 gas. So here, once again, we've got our backscattering. Now it's blue with a peak at 270 meters. So that's the zooplankton. They're adding nitrogen. C to N ratios are decreasing in both suspended and sinking organic matter. You've got this nitrite peak coming from nitrate reduction, and then N2 gas having a peak at the same depth, and here's the qPCR data. So putting it all together, the point is to just see that it's all really happening at the same kind of small depth range. So what does this mean in a slightly bigger picture? It means that there's spatially separated zones of N2 production in the oxygen deficient zone coming from different organic carbon sources. So there's this peak at the top, nitrogen gas peak at the top, which is from sinking particles, and there's also a secondary chlorophyll maximum there, which I'm not talking about here today. But then in the middle, we have zooplankton, but then there's the oxygen deficient zone just keeps going, and the oxygen zooplankton aren't migrating down in there. And so there's a whole range of the oxygen deficient zone that has no extra sources of organic carbon and lower biological nitrogen gas. And I'm bringing this up because the ETMP in particular has been expanding vertically, and it really depends a lot. How important that is depends a lot on whether it's expanding at the top or the bottom. Because if it expands at the top, you have more sinking particles to do N2 production, but at the bottom, there's really not that much going on, so it doesn't matter as much. Okay, so I think I have time for just a couple slides about this zooplankton carcasses and N2 production rates. So we did in situ N2 production rates. The reason we want to do this is because every time you bring water up from an oxygen deficient zone, it goes from having no oxygen and then it gets contaminated. Even small amounts of contamination matter, and then people have to go through and bubble it and decontaminate it again. So Rick Kyle decided to just do the whole thing in the water and just never contaminate the water in the first place. And so to do that, we have these traps. They sat in anoxic water for 8 hours. Then the bottom of the sediment trap closed, top of the incubation chamber closed, and this control bottle, which is really a water column. We have a syringe of N15 nitrate, which injects into the incubation chambers, and they incubate for 22 hours, just like in the water. And the top of the sediment trap was still collecting material. And we found a couple. So we have two types of rates. Then we have a water column rate, and then we have these particle rates. But the particle rates are calculated after accounting for the fact that the organic matter was concentrated for 12 hours. And so we calculated the particle rates for 1 liter of water. So in fact, they are almost by definition, they're supposed to be smaller or the same as the water column rates. But sometimes we have these giant particle rates, and that doesn't fit in the theoretical framework. But when we looked at the carcasses, we found that these really high N2 production rates are linked to the zooplankton carcasses. So here in this graph, this is the difference between the water column and the particle rates when the particle rates are really high. And then we have carbon and zooplankton carcasses on the y-axis, and you can see that there are these huge rates, probably because carcasses contain fresh organic matter that is good for bacteria to eat. So in conclusion, zooplankton add organic matter and reduce nitrogen to ODZ by excreting ammonia, potentially DOM, but we didn't measure that here. Fecal pellets and shedding of cells or molts could make particulate organic matter and also carcasses. And this input of organic matter and reduced nitrogen appears to stimulate N2 production. The fact that zooplankton rest at specific depths during the day causes spatial heterogeneity in N2 production in the oxygen deficient zone. And this can have implications to think about when thinking about oxygen deficient zones expanding, about where that's important and where that's not important. And that's it.
Great. Thank you so much, Clara. That was really interesting. Lots of cool things to unpack. Does anyone have any questions? You can either raise your hand, type it in the chat, or just unmute. Mm, not yet. Okay, I have one. I wonder if you, I know this was, specifically, um, one time period, but I wonder if you have any idea of, um, kind of how this changes, temporally, like, if there's ontogenetic migration, kind of how that would
Well,
things.
the ETMP is in the tropics. So it really, there's so much, I think if it was somewhere else, there would be a lot of change, but we really see these signals like the backscattering. You can look at backscattering for lots and lots and lots of cruises. And Daniele had some for like years and years and years from like the 90s and everything. And it's all the same. And I think that's because it's the tropics. It's not the subtropics. The light just doesn't change. The zooplankton are probably going where they're going because of the light level. And since it just doesn't have seasonal changes, they're just going to the same place. Which, eh, yeah,
Bit easy.
I know. I'm sure it's very different with places that have seasons.
Okay.
I got a question. You just said that you have a few years of data now, I mean, in terms of backscattering at least. And you said that kind of the same, but so because of the light levels, zooplankton would select a certain depth, but do you see maybe also a difference because of the expansion of the oxygen deficiency zone?
Well, probably. But we're always going, we keep going back to this same, we have like a favorite station, where you just go back to the same one over and over again, and you act like the whole rest of the place doesn't exist. So there are transects and they're showing that they're expanding, but our particular spot that we're going to, yeah, we just keep going to the same one.
Okay.
Great. Any other questions from anyone? No? Okay, well, thank you so much, Clara.